How many of you have read Eukaryotic translation? How many of you have read Eukaryotic translation? Is there anyone? I do get my question. Is there anyone who has read Eukaryotic translation? That was a simple thing. You have not read. Then again, this is sheer wastage of time for 1 hour. You will not understand. I spent a good amount of time in explaining the basic mechanism. Only thing is there are certain changes in eukaryotic translation that you have to remember. Basic mechanism remaining same. If you have not read, if you have not opened the book, then how can I help you? So you have to pass the examination. Remember, whatever I am telling is the basic thing. These may not come in the examination. More difficult questions will come. Why are you not reading? I mean, we are having a meeting regular basis with the principal and all the other faculty. Your poor performance for the last batch, you know very well. Still, you are not ready. And what I will teach today about eukaryotic translation, I thought that I will simply highlight all those issues which are related to exclusively to eukaryotes, eukaryotic translation. I mean, can anybody say whether ribosomes are present in the free form in the cytoplasm or it is in the bound form? Can anyone tell? Is ribosome present in the free form or in the bound form in the cytoplasm? Or its subunits are present in free form or bound form? Now you don't have that basic information, and I told at least 5 to 6 times this thing in the last class. You know about smooth endoplasmic reticulum and rough endoplasmic reticulum? You remember what is the difference between the smooth and rough endoplasmic reticulum?
রাফ এন্ডোপ্লাজমিক রেটিকুলামের বাইরে রাইবোসোম লেগে থাকে, যার ফলে হচ্ছে প্রোটিন সিনথেসিস করতে হয়, কিন্তু স্মুথ এন্ডোপ্লাজমিক রেটিকুলাম রিপিট সিনথেসিস স্টেরয়েড হরমোনসের স্মুথ হচ্ছে রিপিট সিনথেসিস স্টেরয়েড
থাকে না,
স্মুথ আর রাইবোসোম থাকে না,
এটা ডিফারেন্ট, বাট ওটা ডিসোসিয়েটেড ফর্মে থাকে মোস্টলি। স্মল ইউনিটে লেগে থাকে, লার্জ ইউনিটে ছাড়া থাকে, তাহলে সিনথেসিসই করতে পারবে না। সে at least he could tell something, but that was somewhat a little bit incomplete answer. So if you don't have that minimum information, it will be very difficult to teach you. I mean, I told in the last class, ribosomes are present in the free form and they remain in the dissociated form. They remain in associated form only in two conditions. When active translation is going on, then both the units join together, and when there is regulation, when the protein synthesis has been kept at a halt, maybe due to several reasons, maybe because of non-availability of amino acids or in stressful conditions. These are the situations when ribosomes will remain in associated condition, two units together, because when the both units, subunits are together, no protein synthesis will occur. So ribosomes are present in the cell in two forms. One is free floating, which is in the cytoplasm. These free floating ribosomes causes protein synthesis which are required for the cell, maybe different enzymes, maybe different proteins which are required mainly for the cell, means intracellularly, including some maybe for plasma membrane also. But those which are synthesized in the rough endoplasmic reticulum, where again the small units remain associated with it, only when the active protein synthesis occurs, the large subunit attaches together and forms protein, and these proteins which are synthesized by the rough endoplasmic reticulum are basically made for export, like something through secretory vesicle, like enzymes, extracellular enzymes, digestive enzymes, the insulin, which are not required by the cell but for some other purposes. These type of proteins are synthesized by the rough endoplasmic reticulum, and there is a signal sequence in the peptide chain which will direct the polypeptide chain into that endoplasmic reticulum and Golgi apparatus, and also secretory vesicles and lysosome also, so that they can do the job what for they were prepared or made. So protein synthesis is a very energy intensive or energy consuming procedure, high amount of complexity are involved in it. Therefore, that cannot be wasted. So regulation is a very important step. Deliberately, there is one component of self-directed learning in your NMC syllabus. We are not going to teach you regulation of protein synthesis, but that will be in your SDL, that will be in your seminar where you have to present. And I am telling you, all the topics of seminar, every student must prepare so that the entire syllabus can be read before the third internal examination, well ahead of time. I will call any student to present anything, all the topics, not that whatever will be allotted to you. So this will be basically an exercise that is called self-directed learning. See, if I teach and if you do not read, then even if I teach regulation of protein synthesis, nothing will enter into the brain. So once you know the regulation, then you will be forced to read synthesis translation, basically prokaryotic translation and eukaryotic translation. Remember that thing. So I told you earlier that protein synthesis occurs in the ribosome, mRNA, tRNA, nucleotides, ATP are the basic requirement. Protein synthesis mechanism is basically ribosomes along with mRNA molecule and tRNA molecule, and several initiation factors are involved. Once the initiation complex is formed, then the initiation of protein synthesis starts. And the starting codon is AUG. In case of prokaryote, it is formylmethionine. In case of eukaryote, it is methionine. A tRNA carries the appropriate amino acid through its anticodon arm. It recognizes, places it right at the P site, the first codon, first amino acid, and all subsequent amino acids are placed next to it on the A site. Peptide bond is formed, tRNA is released, tRNA is moved, dipeptidyl dipeptide is formed, dipeptidyl tRNA is moved to the P site, and it moves again, translocates one codon ahead in front of towards the 3' end, so that the next amino acid can be added to it. I told you about 3 sites. If you have not read it, then what should I do? Where ribosomes are synthesized, within the cell. Yes, nucleus. Ribosomes are synthesized in the nucleus. Nucleoproteins are also I mean, they enter from the cytoplasm through the nuclear pore into the nucleus at the rate of 1,40,000 proteins per minute, out of which they basically make the ribosome and 2000 molecules come out. So only 70 times less than what is entering into the ribosome is coming out. In an active cell, actively dividing cell, the number of total ribosomes ranges from 5 million to 10 million. So that much high amount. So they are basically produced mostly in the nucleus or nucleolus or nucleoplasm. If you cannot remember anything, at least tell it is present in the nucleus and in eukaryotes, and then it is transported through the nuclear pore into the cytoplasm. Okay. So if there is a defect in the formation of ribosome, can you imagine what will be the situation? Protein synthesis will basically not occur. This is very, very rare, and a number of diseases are also associated with it. Those who are planning for pediatrics or obstetrics, they must know these things. Because the babies will be born with these type of defects, and that will mimic with several other diseases. So now you try to understand this thing. I will explain the entire topic once again for your benefit, but now in the class itself you try to remember. Only thing is that you have to read whatever you have to read in the book, from the book, that I will not tell you, that will be shown, but it will be written in the book. From that book you will read it. Now note down the salient points here. So ribosomes in prokaryote, it is 70S. Everything is written in the book. You can write down also into the paper so that you can memorize right now. It will be easier for you to read from the book and to write also. See, ribosomes basically size is 70S. 70S means Sefberg unit. S stands for SVED. I told you it is a sedimentation coefficient. So if you have not read it, it is difficult, I am telling you. See, there is a smaller unit, there is a larger unit. In eukaryotes, it is basically 40S, and this is 60S. Now, these two units remain associated with each other through non-covalent interaction, not through covalent interaction. Otherwise, they would have been required to cleave it. So, just like protein folding secondary structure, hydrogen bond, hydrostatic bond, hydrophobic interaction, van der Waals force, all these forces which are common to make a three-dimensional structure or higher order of structure will be the same here, because these units remain dissociated normally. The small units remain dissociated with the larger units. They normally remain dissociated, and mRNA basically remains here. And as I told you, there are three sites in the larger unit. One is P site, one is A site, and one is E site. E stands for exit, it is the exit route for the tRNA. P site is for peptidyl amino acid, peptidyl tRNA. But this is the site only for the initiator amino acid. Remember, both in prokaryote and eukaryote. This A site is for the incoming amino acid. In all subsequent stages, except the first one, all the incoming amino acids will come to this site only, A site. And there will be energy requirement. So, mRNA synthesis, this will be positioned such that the starting codon, that is AUG, maintains the position at P position. And I told you there is a ribosomal RNA in case of prokaryote, in case of eukaryote also, they will make a double-stranded RNA molecule, hybrid, complementarity. So you note down here, see, now if you break these two units down, so there will be basically this 50s unit and 30 unit. But here now today we are concentrating on the eukaryote. It will be divided into 60s and 40s. Now I told you in the earlier class that peptidyl transferase center is located here. That peptidyl transferase contains peptidyl transferase enzyme which is responsible for peptide bond formation, and that enzyme also cleaves after the protein synthesis has taken place over at the termination codon. Okay, so you see here this 23S rRNA, small unit, subunit, basically this was in 16S rRNA, and now we are talking about 18S rRNA. So in the 18S rRNA, we will make a complementary double-stranded RNA dimer here. But the peptidyl transferase center, peptidyl, that ribozyme or peptidyl transferase enzyme will be located in the 20S rRNA. Here it was in 23S rRNA prokaryote, it will be here 28 rRNA. Initiation basically in ribosome prokaryotes, basically translation and transcription are coupled. Transcription, translation are coupled. There is no RNA processing for mRNA. But I told you during transcription class that there is extensive processing of mRNA like 5' capping, poly A tail addition. All these things are required because this is the reason why so many transcription factors are required, initiation factors are required to make a huge complex. Now the consensus sequence where it will have to start. So there is a signed Dalgarno sequence here, a little ahead, 3 to 10 nucleotide ahead of this starting codon. Similar sequence is there for eukaryote, that is called Kozak consensus sequence. Okay, Kozak. Kozak sequence or Kozak consensus sequence. There is again basically AGGA, GGU, GG. I told you, but I will explain it in a bit detail about it, this thing. So this is the sequence you see. Signed Dalgarno sequence is basically a 6 nucleotide, AGG, but in E. coli only U is also attached. It was the E. coli where the experiment was carried out. Similarly, the Kozak consensus sequence is like this. This is the starting codon. You see AUG is the starting codon here, and at R position, there the prevalence of or the number, the chances of having adenine is much more than the guanine, and GCC region is of uncertain significance. Anything can occur. So initiating codon in case of prokaryote, it was formyl methionine and it was a methionine. Remember, there are two tRNA molecules which are specific for methionine. One is for the initiator codon and the other one is for the methionine inside the polypeptide chain. But these two tRNAs are recognized by the same amino acyl tRNA synthetase. It is the same amino acid. So this is the Kozak consensus sequence in for eukaryotic translation. So initiating factor was basically 3 was required, IF1, IF2, IF3, but here the number is almost 10. 10 initiation factors are required. Peptidyl transferase, as I told you, it was located in 23S RNA, and here it is in the 20S RNA. The synthesis rate is quite high, 15 to 20 amino acids per second, in contrast about 2 maximum 10 amino acid per second. The amino acids are added. Post-translational means when the transcription is going on, translation is also occurring. That means translation is also taking place. That's why its name is post-translational or post-transcriptional, you can say. This is post-translational. Means post-transcriptional, sorry, barely post, I have written it wrongly, post-transcriptional modification, not translation. It is my mistake of writing. So antibody can inhibit this thing. There are, in all the books I have seen, only these 3 toxin, 3 names of these 3, that is diphtheria toxin, ricin and puromycin. These inhibitors are actually not used because of the toxicity. Diphtheria cases are not seen because of the DPT vaccine. Diphtheria cases are not seen. Only thing is that its problem was identified when it was occurring, and what is its mechanism of action? That is the reason diphtheria toxin. Ricin and puromycin, these are 2 toxic. They are used for the laboratory study of translation, whether these are acting causing translation inhibition or not. The only thing is that they will stop some biochemical path during the translation. And what biochemical path, that you have to remember for MCQ purposes. So as I told you earlier, there is EFTU in the prokaryote. TU stands for thermo-unstable, write down. TU stands for thermo-unstable. TS stands for thermostable. So basically it is elongation factor, how to characterize it. Similarly, similar eukaryotic analogy like this, they perform almost the same function. So main factor why, how our ribosomes are kept in dissociated form in the cytoplasm, because the main attachment is IF6, also IF3 and IF1 is also responsible. So they, when they remain attached to that larger subunit, they do not associate. So they are kept dissociated. The very basic purpose is to keep them dissociated, because if they associate, they will not be able to synthesize protein. Again, one release factor will be, how they will be dissociated? There is no mechanism. The release factor coming after the protein synthesis over will no longer act. So they have to be kept in dissociated form somehow. So that is the reason this initiation factor basically will keep them apart. Initiation factor were only 3, but here if you see it is almost 10 numbers. And they act in along with each other, one after another, at times together, to make a huge complex in the initiation complex that we will see shortly. And the elongation factor are only 2. One will place at the first one, and the second one will be in the incoming amino acid. That's the purpose of elongation factor. Termination factor, you have seen that in the prokaryotes there are 3. Basically, one is RF1, RF2 and RF3. RF1 is recognizing UGA and RF2 UGG, and after to release either RF1 or RF2, RF3 will be required. Here RF1 is the single release factor. Now one RF1 binds at the termination codon, then RF3 basically acts to cleave it and then dissociate the entire thing. So ribosome reassociation will take place, that will require the help of initiation factor 5. So ribosome will be kept dissociated with a dissociation factor. Ribosome will be kept with a dissociation factor, initiation factor. They will be united. You will see shortly that E1F will be required during the initiation complex. So this is the chart you have to remember. There is a book, in the book it will get the chart. What are the functions of the initiation factor? At least mainly try to remember whatever you can, so that you can write something in the examination. We don't expect that you will write the entire chart. It is not humanly possible. It is not possible to memorize. Understand, and then today you just complete this, both the chapters in your Jambulkar book. It will take about maximum 1 hour time to read. Only thing is that now you concentrate completely here. This was not written in this format in Jambulkar books. But only thing is that it is written schematically, very schematically. See, basically what is done here, 5' end, 3' end, it is getting this nucleotide code, that is codon, is basically converted into amino acid, that is polypeptide chain. Okay. So it is, again I am telling you, it is monocistronic, means only one usually polypeptide chain is produced at a time. But if the cell demands, then mRNA can be synthesized from multiple points as well. So requirements, basically, as you know, requirement of components, ribosomes 2 units, mRNA, tRNA, amino acid, ATP, initiation factor, and all these things will produce activation of amino acid, that is with the help of amino acid tRNA synthetase. Amino acid will be activated, and that activated amino acid will get attached to tRNA. And then protein synthesis will start. At one point of time it will end also, it will be completed, and then post-translational modification will also be done, and protein will be folded in different configuration. So one name, you write down one name, that is chaperone. Chaperone. They basically help in protein folding. You will be surprised to know that in the last examination, one student, I saw him sleeping in one examination during the last period. But when he appeared for the viva, only a single question was asked, and to our utter imagination, he could draw the graph correctly, and the examiner gave 20 out of 20 to him. And that student gave a proposal on bioinformatics on protein folding itself. I am quite happy to see such type of students, that those who sleep in the examination have also that much idea. I told him that how can we imagine that a computer will do a remodeling of protein folding? At least he has thought of it. He has given a project, and this is now with the knowledge of everybody. I wish that some of you give some more things which will be something unique. That will not require anything but a computer modeling of protein folding after the synthesis is complete. It's quite interesting for me also. I do not know. Now, if those who have attended my amino acid class, they will remember that I told you a single letter abbreviation. KDEL sequence once I told. Now, you see the KDEL sequence here. After the protein is synthesized, if the C terminal of that protein contains KDEL sequence, this will enter into the luminous surface of the endoplasmic reticulum. So that will direct. So that is a function of KDEL. So requirements, as I told you, these are the requirements of amino acids. This I am not going to tell you repeatedly. elongation factor, initiation factor and termination factor, energy source will be from two sides. One will be about ATP, that is the initial stage, that is formation of amino acid tRNA, and two units of two molecules of actually basically phosphate or pyrophosphate will be basically high energy compounds. Two high energy compounds will be wasted in making one amino acid tRNA. Now this I have explained in detail. As I told you, cellular location. Again you note down here, it is present in the free form and in association with the ribosomal rough endoplasmic reticulum, but remember in the rough endoplasmic reticulum, smaller unit remains attached to it. When active synthesis takes place, then only larger unit combines with it. Okay. So these are the proteins which are synthesized targeted for transport, export, or within that which will enter into the rough endoplasmic reticulum inside the lumen or to the Golgi apparatus to the lysosome. So this site I told you, remember at least draw this picture, three sites, peptidyl site, amino acid site and the exit site. Now tRNA, this clover leaf structure of tRNA might come as a short note as well. What you have to write down, you have to draw a picture, you have to write down the arms and write down what are the functions of the arm, and make it will be complete if you write mRNA sequence with an example also. Okay. So that activated amino acid will get attached to the amino acid, this amino acid arm here. Anticodon arm is here. tRNA, this arm is actually recognizing the amino acid tRNA synthesis. This arm is properly positioning it onto the mRNA. So the function of the tRNA is ready. So elongation factor, initiation factor I told you it was almost 10 number. Now you see that E1F, it was having 4A, 4B, 4G, 4E, 4F. So all the initiation factors will be attached to that small unit. So that will be a huge structure. Then only a larger unit will get attached to it, and after that all the units will also be dissociated, so that both the ribosomes subunits remain together. Then the elongation factor will come, one will be there, one alpha will also be there. The two elongation factor, two release factor, main release factor is RF1, I repeat, and to release RF1, RF3 will be required. So steps, at least write down protein synthesis, these 4 steps. First, ribosomal dissociation. In case of eukaryotic, there are 2 steps. One is 43 initiation complex, pre-initiation complex, 48 initiation complex. Then amino acid tRNA, and then the 80S ribosomal complex. That will make the initiation. Then elongation will be basically attachment of the tRNA, amino acid tRNA to the P site, and then subsequent amino acid will come, it will form a dipeptide, and the chain will continue to grow, and the ribosome will move from one codon to the 3' end to make or to add one more amino acid to it. That will be elongation, and then at the end termination codon will come. So newly synthesized protein will be released along with all the factors, so that the entire cycle can repeat. Only that dissociation factor will remain attached to it later. So this is amino acid tRNA synthetase. Amino acid will be activated. You see here PPI, that is 2 high energy phosphate bond is required. So because high energy it is already activated, no energy is required for peptide bond synthesis. ATP will be required during initiation phase, during elongation phase, and during termination phase. তোকে আমি গোটা প্রেজেন্টেশনটা দিয়ে দেবো, এই ছেলে গোটা প্রেজেন্টেশনটা দিয়ে দেবো, মুখস্থ করে নিবি, ব্যাস। আমি নিয়ে কি করব, আমার কি কাজ আছে? শোন, বসে বসে মুখস্থ করে দিয়ে দেবো, এখন প্রেজেন্টেশন দিয়ে দিচ্ছি, নিয়ে কি করবো আমি? আমি খাবো? তোদের জন্য এনেছি, একটু শোন বসে বসে, নিয়ে আমার প্রেজেন্টেশন চা আমি দিয়ে দিচ্ছি, এখন দিয়ে দিচ্ছি, কি করবো আমি নিয়ে গিয়ে? একটু শোন ভালো ভালো, এদিকে তাকা, এই এদিকে তাকা, এদিকে তাকা, অ্যামাইনো অ্যাসিড এসছে, ওকে টাকা, কোনো অসুবিধা নেই, আমি আছি তো, আছি, এর পরে আছি, দরকার পড়ে শিখিয়ে যাবো, যাবো, লজ্জা পাওয়ার কোনো দরকার নেই। এখানে টাকা বেসিক্যালি এটিপি লাগছে, ওকে, এটিপিটা ভেঙে যাচ্ছে, অ্যামাইনো অ্যাসিড টিআরএনএ সিনথেসিস, নামগুলো শুধু মুখস্থ কর, এখনই বসে বসে মুখস্থ কর, ঘরে আর পড়তে হবে না। এইটা করার পরে এটিপিটা ভেঙে যাচ্ছে, দুটো ফসফেট আর এএমপি থেকে তৈরি হলো, এটিপি থেকে দুটো ফসফেট চলে গেল, তাহলে এএমপি তৈরি হলো, এনজাইমের সাথে অ্যামাইনো অ্যাসিড লেগে গেল, অ্যাক্টিভেটেড অ্যামাইনো অ্যাসিড তৈরি হলো, সেই অ্যাক্টিভেটেড অ্যামাইনো অ্যাসিডটা টিআরএনএর সাথে অ্যাটাচ হলো, ওকে, এইবারে এই অ্যাক্টিভেটেড অ্যামাইনো অ্যাসিড টিআরএনএ তৈরি হয়ে গেল। এই অ্যামাইনো অ্যাসিড টিআরএনএ তবে অ্যাটাচ করতে পারবে, তাছাড়া পারবে না। ফ্রি টিআরএনএ গিয়ে কিছুতেই ওটা জায়গা, ওই জায়গা থেকে কোনোভাবেই প্রেস করতে দেবে না। বোঝা গেল ব্যাপারটা? ফ্রি টিআরএনএ তো ঘুরে বেড়াচ্ছে সাইটোপ্লাজমে, ফ্রি টিআরএনএ গিয়ে কিন্তু ইনিশিয়াল কোডনে যেতে পারবে না, তো অ্যামাইনো অ্যাসিড আরএনএতে যেতেই দেবে না, ঢুকতেই দেবে না অ্যাক্টিভেশন ছাড়া। So tRNA needs to be charged, then only it will recognize. Do you understand my point? Free tRNAs are available in the cytoplasm, but until and unless a proper amino acid is actually placed with the, which will match with the correct sequence, then only it will be allowed. Otherwise, it will be rejected simply. Extensive proofreading is required for eukaryotic translation. Because an abnormal protein, if there is an abnormal insertion of a amino acid at a particular, basically very specific position, it will not cause the protein to function. There will be abnormal folding. Very crucial step. So proofreading, extensive proofreading mechanism is there. And this is also done by the large subunit. Okay? মুখস্থ হয়েছে? মুখস্থ করে এখানে। Just try to memorize. At least this step you must not forget. এখানে কি হলো? রাইবোজোমাল ডিসোসিয়েশন হয়েছিল, এবারে 43S rRNA তৈরি হলো। 43S rRNA কি হবে? tRNA কমপ্লেক্সটা মনে করো। Try to remember tRNA complex. tRNA complex was basically three things. One is activated amino acid tRNA, একটা IF2, ওখানে IF2 ছিল, এখানে EIF2 হবে, আর বেসিক্যালি GTP। এটা দিলেই tRNA কমপ্লেক্স। এই tRNA কমপ্লেক্সের সাথে যদি ওই ইনিশিয়েশন ফ্যাক্টর 1, 2, 3 অ্যাটাচ থাকে, তার সাথে সেটাকে বলে 43S প্রি ইনিশিয়েশন কমপ্লেক্স। দিস 43 ইনিশিয়েশন কমপ্লেক্স, প্রি ইনিশিয়েশন কমপ্লেক্স, এর সাথে যখন এমআরএনএটা যুক্ত হয়ে যাবে শর্ট স্মলার ইউনিট, তখন এটা 40S rRNA তৈরি, 40S রাইবোজোমাল ইউনিট হবে, দ্যাট ইজ কল্ড ইনিশিয়েশন কমপ্লেক্স। রিমেম্বার, 43S ternary complex getting attached to the tRNA. 3 factors along with the tRNA. That is forming 43S rRNA, which is attached to the smaller subunit. When mRNA is attached to it, then it becomes 40S initiation complex. And that structure will now form the protein. So formation of 80S initiation complex. এতক্ষণে হলো। বোঝা গেল ব্যাপারটা? এই দেখো ভালো করে, ডিসোসিয়েশন ছিল 6টা দিয়ে ডিসোসিয়েটেড অবস্থাতে আছে, এখানে এ লাগানো আছে, 1টা লাগানো আছে, সুতরাং ডিসোসিয়েটেড অবস্থাতে আছে। এখানে দিস ইজ দ্য টার্নারি কমপ্লেক্স, এই টার্নারি কমপ্লেক্স এলো, এর সাথে ফ্যাক্টরগুলো যোগ হলো। E5টা এখানে আছে, দেখা যাচ্ছে এখানে, এখানে এই ইউনিটটা আছে, এটা 6টা এর সাথে অ্যাটাচ করা আছে, 3টে সাইড এখানে আছে, লার্জার ইউনিটটা। এই ইউনিটটা 43S প্রি ইনিশিয়েশন কমপ্লেক্স, এইটার সাথে এটা যুক্ত হবে, যদি যখন যুক্ত হবে, when it will be attached, then it will form 43S initiation complex. Again, because this unit will have a Septemberg unit of 43, that's why its name is 43S. এইবার 48S ইউনিটটা তৈরি হবে যখন এমআরএনএটা ওই স্ট্রাকচারের সাথে পুরোপুরিভাবে লেগে যাবে। এই স্ট্রাকচারটা যখন হবে, বেসিক্যালি এই স্ট্রাকচারে যখন হবে তখন সেইটা হয়ে যাবে 48S ইনিশিয়েশন প্রি ইনিশিয়েশন কমপ্লেক্স। ওকে, তারপর যখন এইটার সাথে এইটা অ্যাটাচ হবে লার্টা ইউনিটটা, সেইটা হবে গিয়ে তখন ওই সাইডটা চলে আসবে, তিনটে সাইডে চলে আসবে। বাট এইবার দেখ এখানে ক্যাপ বাইন্ডিং প্রোটিন একটা আছে, দেখ 7-methyl guanosine capping is unique for mRNA and poly-A binding, poly-A tail. তা 7-methyl guanosine বাইন্ডিং একটা প্রোটিন একটা দরকার আছে, ক্যাপ বাইন্ডিং প্রোটিন বলে। এই ক্যাপ বাইন্ডিং প্রোটিন হচ্ছে কি 4F, 4Fটা কমপ্লিট হচ্ছে তিনটে ইউনিটের সাথে অ্যাটাচ হচ্ছে। ওকে, a cap binding complex protein complex is the actual rate limiting step, just like cholesterol synthesis. HMG reductase is the rate limiting step. Similarly, the rate limiting step of translation in eukaryote is this one, recognition of the mRNA cap by EF4. Is a 4F, E1F4 or 4F meaning is same. It is a short form of E1F4, E4F. So cap binding protein is there. This is cap binding protein. This is poly-A binding protein. PAB means poly-A binding protein. So দুটো বাইন্ডিং প্রোটিন দিলে পুরো স্টেবিলাইজ করলো জিনিসটাকে, এবার এটা স্টেবিলাইজ করা হলো, পজিশন করা হলো, যাতে ওরা এবারে সিনথেসিস তৈরি করতে পারে। এইবারে 40S mRNA, যেটা আমি এতক্ষণ ধরে বলছিলাম, যে এই স্মলার ইউনিটটার সাথে এই সমস্ত অ্যাটাচমেন্ট হলো, তার সাথে mRNA অ্যাটাচ হয়ে গেল। ইউ সি দ্য হেয়ার, ইট AUG ইজ হেয়ার, দ্যাট মিনস ইট ইজ অ্যাটাচ টু দ্যাট প্রোজাক কনসেনসাস সিকোয়েন্সে, এখানে এসে অ্যাটাচ করা আছে। এটাকে একটু মুভ করতে হবে, তবে AUGটা এখানে আসবে। এটা মুভ করবে, ওই ইনিশিয়েশন ফ্যাক্টরটাকে নিয়ে আসবে, নিয়ে এসে ইনিশিয়েশন ফ্যাক্টরটা এসে লাগবে, তবে এখানে গিয়ে অ্যাটাচ হবে। এখান থেকে 4Fটা এখানে বাইন্ডিং করেছে, ক্যাপটাকে কানেকশন করেছে, পলি-A বাইন্ডিং প্রোটিন এখানে অ্যাটাচ হয়ে গেছে। এবারে এইখানটাতে AUG আছে। ওকে, এই যখনই এই জিনিসটা তৈরি করবে, এই এটিপি, এই জিডিপিটা ফাংলি, জিডিপি থেকে একটা জিডিপি তৈরি হবে। এই গেল, এইবার দেখে এটা মুভ করেছে, না ইট হ্যাজ মুভড ফ্রম দিস প্লেস টু দিস ওয়ান, আর দিস ইজ বেসিক্যালি লার্জার ইউনিট, দিস ইজ স্মলার ইউনিট, এখানে এলে একটা পজিশন করে দিয়েছে। এবার জাস্ট উল্টো দিকে আমি যেটা ছবিটা আঁকছিলাম সেটা ওই দিকটাতে, এবার ছবিটাকে জাস্ট এখানে বয়ে দেওয়া আছে নিচের দিকে, আর কিছু না করে। এইটা এই পজিশন করলে এই জায়গাটাতে AUG ঠিক এই নিচের দিকে নিয়ে এসছে ছবিটাকে। আমি এই ছবিটা এইরকম করে আঁকা আছে। বাস, আর কিছুই না, গোটা ব্যাপার বোঝাতে পারলাম? হলো। এবার এই দেখো 4টি ইউনিট, 43S, 48S, দেন 80S। এইবার ট্রান্সলেশন এই জায়গাতে একদম অ্যাটাচমেন্ট হয়ে গেছে। এখানে PF বাইন্ডিং প্রোটিন আছে, 4F বাইন্ডিং আছে, পুরো রাইট পজিশনে লোকেট পজিশন করে ফেলেছে ইনিশিয়েটর কোডনটাকে। আর দিস ইজ F-met RNA, tRNA। এইবারে বাইন্ডিং হয়ে গেল এই জায়গাতে পুরোপুরি, এইভাবে বাইন্ডিং অফ অ্যামিনো অ্যাসিড tRNA আসছে। এবার এই জায়গা থেকে নিয়ে এলো, এই দেখো ভালো করে তাকাও, এই দিকটা তাকাও, এই দিকটা EF1 আলফা, এই 1 আলফা এই জায়গাটা ব্লক করে রেখেছিল, 1 এই জায়গাটা ব্লক করা ছিল আগে। এইবারে এই জায়গাটাতে এটাকে নিয়ে এসে প্লেস করবে, এই প্লেসিং হয়েছে। ওকে, দুটোকে যখনই প্লেস করলো তখন একটা পেপটাইড বন্ড তৈরি করবে। ওর রাইট সাইডের কার্বক্সিল গ্রুপ, এই লেফট সাইডে, রাইট সাইডে যেটা রয়েছে তার অ্যামিনো গ্রুপটা, আর লেফট সাইডে যেটা আছে তার কার্বক্সিল গ্রুপটা, এরকম জিনিসটা তৈরি হবে। আগের দিনে বললাম এর কার্বক্সিল গ্রুপটা আছে এইদিকে, আর এর অ্যামিনো গ্রুপটা আছে এইদিকে। এই পেপটাইড বন্ডটা তৈরি হলো, দুটো অ্যামিনো অ্যাসিড, কিন্তু এর জন্য কোনো এনার্জি লাগবে না, অলরেডি এনার্জি ওর মধ্যে আছে অ্যাক্টিভেটেড। এই প্রসেসে জিটিপিটা রিলিজ হয়ে গেল। এই জিটিপিটা ভেঙে যাবে, আর সব করে ইনিশিয়েশন ফ্যাক্টর ডিটাচড হয়ে যাবে। এই যে আমি আবারও বলছিলাম যে 48 এসে এই 28 এটার মধ্যেই পেপটিডিল ট্রান্সফারেজ এনজাইমটা আছে, আলাদা করে কিছু লাগবে না। সো রাইবোজাইম ইজ দেয়ার ইন দ্য লার্জার ইউনিট ইটসেলফ। ওকে, ইট ইজ অলরেডি অ্যাক্টিভেটেড। এই যে নো এনার্জি ইজ রিকোয়ার্ড। এরপর হয়ে গেলে ট্রান্সলোকেট করবে ওয়ান কোডন ahead on the right side, then there will be detachment procedure will start. That tRNA which is attached to it needs to be now made free. That will be recycled in the cytoplasm. So এই tRNAটা এবারে এলো এদিকে, এক্সিট সাইডে চলে এসছে, C2 বেরিয়ে এলো এখান থেকে, tRNA চলে এসছে, আবার ফ্রেশ সাইকেল এটা তৈরি করে নিয়ে আবার ওখানে নিয়ে গিয়ে আবার কোনো ইনিশিয়েশন কোডন হিসেবে কাজ করতে পারবে। এই tRNA ইজ স্পেসিফিক ফর ইনিশিয়েটর কোডন। এই চললো, এই যে তৈরি হচ্ছে, এই তৈরি হলো, একটা সময় আসবে আলটিমেটলি স্টপ কোডনের কাছে আসবে, টার্মিনেশন কোডনের কাছে আসবে। টার্মিনেশন কোডনে যখন এলো তখন এই টার্মিনেশন কোডনের 3টে ওয়াটার মলিকুল একটা প্রোভাইড করতে হবে, কেন ওই যে বন্ডটা আছে অ্যামাইনো অ্যাসিড tRNA এর সাথে, পেপটিডিল tRNA আছে, দ্যাট ইজ অ্যাটাচড, এস্টার বন্ড একটা আছে, এস্টারেজ এনজাইম লাগবে। এস্টারেজ এনজাইমটা বেসিক্যালি হাইড্রোলাইসিস করাবে। দ্যাট হাইড্রোলাইসিস, দ্যাট ওয়াটার উইল বি প্রোভাইডেড বাই RF1, রিলিজ ফ্যাক্টর 1। ওকে, সেই রিলিজ ফ্যাক্টর 1 সে কিন্তু এগেন ওই পেপটিডিল ট্রান্সফারেজ সেন্টার থেকে যে এনজাইম আছে ওই 20S rRNA ওইটাই করবে। দুটো কাজই করছে, বন্ড ফরমেশনও করছে, পেপটিডিল ট্রান্সফারেজের কাজে করছে, অ্যাট দ্য সেম টাইম এস্টারেজ কাম হাইড্রোলাইজ ইউ ক্যান সে, সেই কাজটা করে অ্যামাইনো অ্যাসিডের চেনটা, গোটা অ্যামাইনো অ্যাসিড যা তৈরি পলিপেপটাইড চেন, সেই চেনটাকে রিলিজ করে দেবে, আর তার সাথে আরও বাকি জিনিসপত্র সব রিলিজ করে দেবে। সো নিউলি সিনথেসাইজড RNA বেরিয়ে গেল, RF1 এখানে এলো, RF3 অ্যাটাচ করলো, জিডিপি থেকে আবার জিটিপি একটা এনার্জি লাগবে, যেখানে যাই কিছু করতে যাবে এনার্জি একটু লাগবে, তাহলে ইনিশিয়েশনে একটা এনার্জি লাগলো, ইলংগেশনে একটা সময় লাগলো, আর টার্মিনেশনের সময় একটা এক্সট্রা এই জিটিপি লেগে যাবে, আর ওই ইনিশিয়েশন ফেজে দোকানে লাগবে। ওকে, টার্মিনেশন। এই রেগুলেশনটা খুব কমপ্লিকেটেড আছে, তাহলে এই রেগুলেশন করতে হবে, হোয়াই? বিকজ at what is the need of the cell, depending on the need of the cell, because once the protein is synthesized, that protein have to be kept for some time in the cell, otherwise there will be sheer wastage of energy and time, sheer wastage. I mean, again to start, so the life of a protein, during my protein class I told you the life of a protein just like a human being. So there are various proteins have various lifespans. So once the protein part is over, then the protein will be broken down into amino acids, and if they are within the cell, they will be recycled back. All the amino acids will be again utilized for again new protein synthesis. It can be a structural protein, it can be a functional protein, it can be a small peptide, it can be large peptide as per need of the cell. So phosphorylation, just like your glycogenolysis, if you remember glycogenolysis, phosphorylation of serine, threonine, these are basically regulators of many biochemical mechanisms. So here also again phosphorylation is one of the primary regulator of protein regulation. This you will tell in your seminar class, in the self-directed learning class. So basically I am telling you this thing. So phosphorylation of EF2 regulation you will tell in detail. This may come as a short note, just like post-transcriptional modification of RNA, post-translational modification of protein. Remember, two things are there. One is proteolytic cleavage. First job is to remove the initiator mRNA. With that initiator protein, amino acid, that protein is common for all, that amino acid is common for all proteins. So that will be required to be removed. At times a signal peptide is there, which is required to direct that particular protein to a particular location. So that signal peptide may be also required. And some extra proteolysis also will be there. So in zymogen granules, you have seen that trypsinogen gets converted into trypsin, or pepsinogen is getting converted into pepsin because of the cleavage of that extra amount of peptide chain. Okay. So covalent modification are a number. This is the insulin chain that you have read several times. Proinsulin, and then AB chain linked to basically disulfide bond. That structure you try to remember. So that is again a post-translational modification. This structure has been shown to you, and I have read it in the book also. So zymogen to active form again is basically a post-translational modification. All these things, these points, try to remember all these points only. Glycosylation. Glycosylation can take place while the protein is in the endoplasmic reticulum. So it can be at one place, it can be at multiple places. It can be O glycosidic bond, it can be N glycosidic bond also. Disulfide bond. Again, addition of phosphatic groups, because the protein has to be made after all functional. So post-translational modifications are done to make the protein functional, number one. Number two, it gives stability to the protein and its actual purpose, function can be executed. So hydroxylation of proline and lysine, remember it is required. Hydroxylation of proline and lysine is for collagen synthesis. And then gamma carboxylation, basically it is required for clotting factors. Gamma carboxylation of clotting factors. Phosphorylation, you know, glycogen phosphorylation, glycogen synthesis, phosphorylase, dephosphorylase, and glycogen synthesis also. Predylation and acetylation, even methylation of DNA, histone proteins. These are also comes under the covalent, means it will be very difficult to cleave. Proteolytic cleavage is also basically a covalent bond that requires an enzyme. So amino acid structure of collagen synthesis, I told you, this is triple helical structure. Post-translational modifications are done by hydroxylation and proline and lysine, done by basically vitamin A, vitamin C and copper ion. Lysyl oxidase is the enzyme. Okay. So translation of protein machinery, you know all these things. I am not going to tell once more, everything has been told. So inhibitors of protein synthesis, these three things are written in your book. Try to memorize these three things only, not more than that. Puromycin is basically tyrosine tRNA. So in place of tyrosine, puromycin will get attached to it. So it will, protein synthesis will not be completed. Similarly, cycloheximide, it inhibits the 60S ribosome. If 60S ribosome is inhibited, then this association, that unit will not function. Resin, it activates the 20S rRNA. The 20S rRNA removed means one small unit will not be attached to mRNA. And the diphtheria toxin, ADP ribosylation of elongation factor 2. If it is the elongation factor 2, it gets attached to something else, the elongation factor 2 will not also actually act. So one important point about this is not discussed. Mitochondria is also having DNA and it is having a DNA synthetic machinery altogether all similar line, just like protein synthesis within the cytosol. Only thing is that some proteins are exported into the mitochondria to make the ribosomes there to make its protein synthesis. So mitochondria synthesizes proteins which are required only for the electron transport chain, a limited number of proteins. Main importance is somewhere else, I am telling you. So we know all diseases which are linked to X autosomal dominant, autosomal recessive. Only a limited number of diseases are there which are of inherited mitochondrially. These are called mitochondrial inheritance. And remember, we all get mitochondria from our mother. Cytoplasmic division is there, but during that cell division, mitochondrial DNA comes from the mother. Now, this is of paramount importance when there is a disputed paternity. Who is the biological mother of that particular child? Now, it cannot be said so easily. Now, if there is a mitochondrial DNA is analyzed and if there is a matching between the child and the mother, then that is the biological mother of that child. So, it is having a biological forensic implication. So, mitochondrial inheritance can be, it is due to mother just like an X-linked disease. So, X-linked disease, mother is the carrier and son is the sufferer. Same thing will happen here, and that is the most unfortunate scenario for the mother. Mother herself not knowing that she is diseased or giving birth to a baby for which she is responsible. Very serious, very pathetic condition. So, you remember mitochondrial inheritance, only a limited number of diseases are there. Remember only one thing I will tell you. Only one disease you remember. Liver hereditary optic neuropathy, only one disease, remember, only one disease. At least you will be able to tell. Liver hereditary optic neuropathy, LHON, only one name you write down, remember. Others if you cannot remember, doesn't matter. Remember one thing only. Or as mitochondrial myopathy, it will be easier to remember. So, this is the mitochondrial machinery. There are 100 copies of mitochondria. In the mitochondria, there will be mutated version, there will be normal version. So, this is called heteroplasmy. One term you note down, heteroplasmy. Just note, this is the last slide. So, you note down one term, heteroplasmy means both a combination of normal mitochondrial DNA and a mutated RNA DNA, mutated mitochondrial RNA. So, a baby can get a normal mitochondrial DNA, so there will be no disease. If by chance that mutated DNA is transmitted to the baby, then only he will suffer. Okay. So, the daughter cell may inherit mutant or normal DNA. এই লাইনটা লিখে রাখো ভালো করে mitochondrial DNA is inherited from the mother. So, in evaluation, during evaluation, who is the actual, sufferer, father or the mother, accordingly genetic counseling will be done, and as I told you, for forensic purposes. It can happen, it has happened, I am telling you. One forensic expert told in our conference that there is a dispute. I told in the earlier batch, one thing I am telling you, you try to remember, childless couple usually try to adopt a baby, usually taken from the orphan. In course of time, that grown up child knows that they are not my biological parents. Legal inheritance is a big issue. So, that court case was there, and ultimately it was proved that she was not her biological mother, and that they admitted after the forensic test was done. So, this is the role of a medical person who knows the science very clearly. Under pressure, if you do, that is altogether a different picture, and there will be severe pressure on this also, because they do not want to disclose. I know also a family. Every time they kept on telling, but never tell that we have adopted the baby. It's a sorry state. But they are childless couple. What can I do? I told, I know, why should I tell? The baby told the mother that I am not looking like you. I don't think that I am your child. When she was about 11 years old, and then the mother told me that, Dada, please do not ever disclose that we have adopted her. So that's a peculiar situation. You have to manage as a medical person accordingly. Okay, with this I conclude. You just go and read, and tomorrow I will take 4 more classes I will take, and then goodbye.